A Carnegie Institution for Science audio press release for October 12, 2010. Scientists watch cell shape process for first time. Researchers for the first time have observed in living plants the birth of microtubules that create the cytoskeleton, which is the scaffolding that provides structure and ultimately form and shape to the cell. These fundamental results could be important to agricultural and other research. David Earhart of the Carnegie Institution for Science's Department of Plant Biology is co-author of a paper regarding the research, along with colleagues from the NARA Institute of Science and Technology. All plant and animal cells depend on microtubules, which are molecular rods built from the protein tubulin. When organized into specific configurations, the microtubules organize the cell and generate forces needed to support cell shape, movement, and cell division. Animal cells separate their chromosomes during cell division by organizing the microtubules network from centrioles. A big mystery is how plants, which do not have centrioles, organize their microtubule network. Understanding these mechanisms of molecular organization is a primary goal of cell biology. The Earhart lab previously found that individual microtubules in plant cell arrays are born at many locations along the inside of the cell membrane, where they are detached from the sites of birth and move along the membrane to interact with other microtubules. A primary challenge for investigating the molecular basis for these processes has been visualization of the protein complexes that give birth to new microtubules. The scientists met this challenge by tagging a component of these complexes known as nucleating complexes with multiple copies of a fluorescent protein derived from jellyfish. When introduced into plant cells and visualized with highly sensitive microscopy, this tagged protein permitted the researchers to observe what happens as the microtubule array is being built. In centrosomal arrays, nucleating complexes give rise to a star-shaped array centered near the nucleus. By contrast, the cells in this study were distributed at the cell membrane. They were primarily located along the sides of other microtubules, an association that was correlated with their activity. So, microtubules appear to be important for locating and regulating their own formation proteins. The investigators observed that formation complexes frequently did not remain in place after creating new microtubules, but left, presumably to go through a new cycle of microtubule creation at a new location. The scientists hypothesized that liberation of the complexes from mother microtubules might be related to the mechanism of daughter microtubule detachment from origination sites. The investigators introduced their fluorescent probe into a mutant which lacked the protein catenin, which cuts microtubules into pieces. Catenin is named for a Japanese word for sword. Without the cutting protein, the daughter microtubules completely failed to detach from their birth sites, and tagged formation complexes remained at the base of the daughter microtubules. This research is the first to witness the dynamics of individual gamma-tubulin complex processes, which are fundamental to every plant and animal. The research could help understand basic mechanisms underlying crop plant growth and development, and could have implications for understanding the process of acquiring cell shape and function of human cells. The research was published in the October 10, 2010 early online edition of Nature's Cell Biology. This is John Strom for the Carnegie Institution.